Hey, welcome back. Today we're going to go through Histology 2, and we're going to start with the lymphoid organs. Buckle up, it's going to be a long one. So, of course, it plays a role in immunity. There's the two different types, innate and the specific or cell-mediated, and that's adaptive. There's two different kinds, naturally and artificially acquired. Lymphoid organs are going to be made of connective tissue and the lymphocytes, and they are connected by the blood and lymphatic vessels. So the primary lymphoid organs, they start with basically in development. So all of your immune cells are going to be made in the bone marrow and the thymus when you're young. And then as you age and mature, the secondary lymphoid organs will kind of take up the slack and contribute to the lymph and immune cells that are made by the primary lymphoid organs. So the malt in the lamina propria is going to be a part of the alimentary canal, the respiratory, urogenitory, and the digestive system. And the malts aggregate in certain places, such as the tonsils and the pyre patches, along with the variform appendix. So here's the pyre patches. They lie in the lamina propria. And here's the lymph nodules. We'll go through that. So this, during development, the stem cells come from common progenitor cells, also known as colony-forming units. And those will differentiate into myeloid and lymphoid stem cells. And of course, the myeloid and lymphoid stem cells have different lineages. The myeloid stem cells will give rise to the traditional leukocytes, along with the mast cells, the megakaryocytes, and the red blood cells. In the meantime, the lymphoid stem cells will give rise to B and T cells. The B cells, creatively named, are called that because they mature in the bone marrow and are released as B lymphocytes. T lymphoblasts instead, they are produced first in the bone marrow, and then they travel to the thymus, and then they become the thymocytes. So the T lymphoblasts from the bone marrow enter the cortex of the thymus and make the cortex appear darker. So these are the stem cells. They're far larger. Look here. Here they are. And they are they have like a lot of granules in it. So in the thymus, there's something called thymic education, where the thymocytes differentiate into the mature immunocompetent T cells, along with the CD4 helper cells, the CD8 killer T cells, and the T regulatory cells. So what does that mean? So these cells technically can work. They can attack any antigens, but they haven't been involved in the positive and negative selection yet. So they have a lot of specific cell markers, but they haven't been exposed to anything. So they're called naive. So naive T cells are immunocompetent and mature, but they can't recognize a specific antigen yet. So they have all of these surface markers, but they haven't been able to specialize yet. Kind of like, you go to college, you have all this knowledge now, and then by the time you graduate, you kind of narrow down all of your studies into your specific field. So these naive T cells need to circulate and be exposed, and that is called the antigen challenge, and then they are activated. After activation, it can clone itself, and then that can recognize the antigen, this particular antigen, as an effector, which is like a killer T cell, or a memory T cell. So the thymus itself is a lymphoepithelial organ, and it comes from the third pharyngeal pouch during the 6th week of development. Thymocytes, of course, come in from the bone marrow between the weeks 7 to 14 of development, and then they proliferate in the cortex. See how dark it is right here? After birth, the thymus is fully formed and is active in the T cell production until puberty, and then it goes through an involution. So involution is when the thymus quickly actually becomes almost non-functioning. It's still cranking out a couple T cells, but it's not nearly as much as pre-puberty. And then the tissue of the cortex and the medulla are like replaced by adipose tissue. So the thymus is made of vascularized connective tissue, and eventually that is replaced by the fatty tissue. And look here, the septa come in and divide it into lobules. The lobules will have the cortex here and the medulla in the center, and they're different because the thymocytes are in different stages of development or maturation. And all of this is surrounded by a capsule. Okay, so there's a bunch of macrophages inside, and then the parenchyma is made out of the thymocytes and the macrophages. Parenchyma just means what is the active form or active units of an organ, and the stroma is just like not just the connective tissue, but the surrounding tissue that supports the organ. So it's not like the active part of the organ. The cortex is made out of the thymocytes, macrophages, and thymic epithelial cells, and that's going to be stained basophilic. Here's the cortex, nice and dark. Here's the medulla, far lighter. And the thymic epithelial cells in it were going to have large euchromatic, so very dark purple staining, nucleuses, eosinophilic cytoplasm, so it's going to appear pink, and keratin filaments, also known as tonofibrils. While in the cortex, we're going to have 3 types of thymic epithelial cells. The matured thymocytes will leave the medulla by going through the venial walls, and like here, cortex, and they're just gonna like here's the venial, so it's gonna like just diffuse through and go straight into circulation in order to go through the antigen challenge. The thymus itself has no afferent lymphatic vessels, so there's nothing going in. There's no lymph going in. So type 1 TECs are going to be squamous cells, and they're joined by tight junctions, and the basic function is to shield and isolate the cortical capillaries from the connective tissue, and they rest on the basal lamina. So the point of this type 1 cell is to make the blood thymus barrier. So the basal lamina of type 1 cells, along with the capillary endothelium and the perivascular sheath, containing the macrophages. So here's your macrophage, here's your T cells, here's your type 1. See, it surrounds the capillary endothelium is right here, and it surrounds all this basal lamina. So there's like a bunch of walls, barriers to the blood, in order to prevent the immunothymocytes from unregulated exposure to antigens. So remember, they have a bunch of all these like specific cell markers, but these cells aren't mature enough to, or they haven't been exposed to something particular in order to like specialize. So if they are exposed a little bit too early, it might lead to autoimmune reactions because the cells aren't mature or specified enough to recognize what is an outside antigen and what is like, an internal cell signal. Here are your reticular cells. Here's your blood vessel, and surrounding it is going to be the type 1 epithelial, thymic epithelial cell. Okay, so type 2 are the stellate cells. Those have the tonofilaments, and they're joined by the desmosomes, connecting them all together. And they make up like a cytoreticular network that supports the developing cells, and these will secrete thymosin, thymopoietin, and cytokines. These particular cell signaling molecules provide an environment that is conducive to help the T cells develop. How, honestly, science isn't quite sure yet, but we just know they do help with the development and the maturation. So the type 3 cells are going to be squamous cells that make up the cortical medullary barrier, and basically it's just like a additional little barrier to the type 1 cells, and that will prevent a mixture of the mature and immature cells. So type 4, the medulla is made of more mature cells, and they have types 4, 5, and 6. And those don't have a blood thymus barrier, of course, because they're now mature, they should be able to leave. And the type 4 cells make the second layer. Type 5 cells are stellate cells, so these are more support cells. Type 6 make the Hassel corpuscles, which are aggregates of flattened cells, and those secrete the cytokines that control the dendritic cells. And by dendritic cells, I mean the dendritic cells a part of the immune system, not the dendritic cells a part of the axon and dendrites. So they have factors that promote the differentiation of regulatory T cells for peripheral immune tolerance, and they help supplement the central immune tolerance that develops in the thymus. But eventually they degenerate and are filled with keratohyalin granules and cytokeratin filament during the involution period. And regulatory T cells will prevent excessive response to antigens. People who lack a decent regulatory T cell response, or they just have a deficient amount of it, have excessive response to a lot of allergens. So think peanut allergies, think people who are allergic to like sunlight. So they have a very excessive response. Positive and negative selection are part of the central immune tolerance, and that is, let's define central immune tolerance. It's basically how the body learns how to tolerate its own antigens, its own cells, its own cell markers. So, thymic cell receptors are going to be the surface protein complexes that only react with the MCH and HLA receptors, and these work as markers. So MHC1 are on all cells, and MHC2 are only on the macrophages. So the positive selection makes sure that the T cells have functional TCR in order to recognize our own MCH1. And the detective cells that don't recognize the antigens are, of course, disposed by the macrophages. And there is negative selection in the medulla. And by that means that the defective cells have too many receptors and no strong recognition, so it can cause an autoimmune response. So positive, make sure that they work. Negative, make sure that they don't have too many receptors. Okay, so the secondary lymphoid organs are going to be made out of a lot of type 3 reticular collagen fibers, and they have a lot of antigen-presenting cells. So the plasma cells, follicular dendritic cells, and the reticular cells. And they're characterized by these lumps of cells, also known as a lymphoid nodule. And they're different from the nodes because they're non-encapsulated. As you can see, there's no capsule around it. There's no connective tissue. And they're not permanent, so they're transient. These pop up when you have an infection. And they're not the ones that are palpable. Those are your lymph nodes. But these are small, microscopic little nodules that pop up in just not just isolated not just isolated little places like a cut on the skin or anything, but these kind of pop up everywhere in the body, as you can see in future lectures. So here they're particularly common in the intestines, of course, because we eat, and there's plenty of things that we need to digest and make absolutely sure that the bacteria inside are going to, whatever we've eaten is going to die and not infect us and give us food poisoning. So there's a lot of lymphoid nodules right under the epithelium of the alimentary canal, really, along with the digestive system. So the primary lymphoid nodules are B cells that haven't been exposed to antigens yet. and they make a big aggregate, and they do not have a germinal center. So here's your primary lymphoid follicle. And they are found in the lamina of the mucosa, and they are the first line of defense in the system that opens to the outside. So that's like the alimentary canal, the oral canal, the nasal canal, nasal cavity, the rectum, and they're sources of the B cells making the immunoglobulin A. And then they become the secondary lymphoid nodules after the B cells have been exposed and bound by the T helper cells. See here, here's your germinal center, here's your follicle. So the lymphoid secondary nodules will have the germinal center and a mantle. The germinal centers are going to be active and proliferating B lymphoblasts, and then the naive, non-proliferating B cells, so they've already been matured, and they're going to be pushed into the periphery, that's why they're darker, and become the plasma cells. And of course, there are a bunch of macrophages inside to remove the defective cells. And the mantle is going to be made out of the terminally differentiated B cells, which have a small dark nuclei, which makes the mantle darker. Lymph nodes are going to be mostly in the joint areas and large vessels, and they play a role in filtering lymph, along with making antibodies. So lymph will enter through the convex site and via the afferent lymphatic vessels. So it goes from the wide end, circulate, and then leave via the efferent lymph vessel. They all come together into one big one. There is going to be a whole lot of white blood cells in it, and they're the kidney shaped. So they have the cortex, the paracortex, and the medulla. Problems with lymphatic cancer is that if something grows in here, it's going to erode the structure and enlarge the lymph nodes. And the encapsulated cell structure is going to be filled with the white blood cells. And since it's encapsulated, it's just going to grow, and then it bursts, and then it can spread via the lymph. So the cortex is made out of reticular fibers and cells, along with the sinuses and nodules. The subscapular, so right under the capsule, get the sinus, they get the lymph containing all the free antigens, white blood cells, the antigen-loaded APC, and that all comes in, and then inside it branches as the cortical sinuses. And then the lymph will go through the discontinuous endothelium into the tissue and just kind of circulate in through to reach the lymphoid nodules. So it goes from outside, you go, and it circulates all in. So the paracortex doesn't have any lymphoid nodules, and they're only made out of T cells. They come in they come in from high pressure endothelial venules, and they are the main site of B and T cells from the blood going into the lymph nodes. And these are only in the secondary lymphoid organs. So it's not in the spleen, and it's not in the bone marrow. There's a bunch of surface molecules, and those help the white blood cells move inwards. The medulla, it's going to be the dark spots, and they're going to be branched extensions of the lymphoid tissue of the paracortex. So there's communication there. And they have the B, T cells, and the plasma cells, and those are separated by sinuses. And the medulla sinus is continuous with the cortical sinus, and they all converged at the hilum, which is kind of the middle of the concave part. And those are filled with macrophages and neutrophils. As you can see here, here's from the outside. So the cortex, that's B cells, and then you go inwards to T cells, and then B and T cells. Here's the medulla, and it all goes into the hilum. Paracortex full of T cells, cortex full of B cells. It's all going in. So the tonsils are made in the oral cavity in a ring. So there's the lingual, the palatine, the pharyngeal, and adenoid. And those make the immunoglobulins by the B cells. The palatine and lingual tonsils are covered by non-characterized keratinized stratified squamous epithelium, while the pharyngeal tonsil is covered by pseudo-stratified columnar ciliated epithelium, and those have thin capsule. So the pharyngeal one has the pseudostratified and columnar ciliated epithelium in order to move materials down the throat, right? The mucosa are going to have the invaginations, but no crypts, which differentiates it from the mucosa of the intestines, and the palatine tonsils, because the palatine tonsils also have tonsillar crypts, which on the inside have the white blood cells. Okay, so this is really common, just having infected tonsils, and usually that's just treated with a tonsillectomy. Usually it's the palatine tonsils, but all the other tonsils are subject to infection as well. But usually it's just the palatine tonsils, just the ones that you can see on the sides of the throat. So the spleen is going to be the most important lymphoid organ because it defends from blood-borne antigen, and it's the only organ that does so, because it's the only organ that filters the blood. So it makes the antibodies and the activated white blood cells, and it destroys the old white blood cells and stores some of the blood, but that's not one of its main functions. Remember when we were talking about sickle cell anemia and how that can lead to splenomegaly? It's because of this function. The sickle cell anemia will overwork the spleen, and of course the sickle shape will also build up and cause blockages, and that will pierce the spleen and making it in hypertrophy. Or the correct term is really hyperplasia of the spleen. So it has something called the splenic pulp, which is really important, but we'll go through that in the next slide. This has the capsules, and the capsules will extend into the splenic pulp, and the trabeculae of it will carry arterial branches into the pulp, and the largest trabeculae will, of course, originate at the hilum. They have branches of the splenic artery and nerve, along with the lymph vessels, the trabecular vein, and they will all kind of merge together into the big splenic vein. Or right here. I've been pointing at all the wrong things. This is the pancreas, this is the splenic vein. Okay, so the trabecular arteries will leave the trabeculae and enter the white pulp. So here's the white pulp, which is, strangely enough, dark in the H&E stains, but that's because they don't contain the red blood cells. But anyway, trabecular arteries will enter the white pulp as central arterioles, and the central arterioles are going to be surrounded by a sheath. And they send capillaries through the white pulp to the small sinuses that go and communicate with the red pulp. So that's how the white and the red pulp communicate. And in the red pulp, it loses sheath and branch of the penicillar arterioles. And then enlarged spleens, of course, can, as I said before, because of the sickle cell anemia or trauma or infection, it's really problematic because they rupture so easily. The thin capsule surrounding the spleen hardly does any support. It's just there. Okay, so white pulp is made out of the sheath, PALS, and the lymphoid nodules, and it's mostly made out of T cells. Lymphoid nodules are going to be made out of the proliferating B lymphocytes in the sheaths. So the red pulps are going to be made out of the splenic cords, also known as Billroth's cords, and sinusoids. The cords run through the sinusoids, and they have all these cells. And the sinusoids are lined by stave cells, which are important because they differentiate the senescent, or the old and unhealthy red blood cells, from healthy red blood cells. The sinusoids are going to be wrapped in reticular fibers, which form a discontinuous basement membrane in order for the healthy red blood cells to pass through easily. Marginal zone is kind of like the transition zone. There's a lot of active macrophages to, clean up the lymph, to eat the, bacteria, toxins, various other antigens. And they have dendritic cells as well, which activate the B cells to go into, to differentiate into plasma cells. Okay, so there's two types of circulation, closed and open. So the closed flows directly into the splenic sinuses, and then from there goes right into like the splenic veins, while the open flows from the capillaries into the red Pope stroma, and then through the spaces between the stave cells, and re-entering the sinusoids, trabecular vein, and then the splenic vein. Why all of that, kind of a roundabout way of going through it is so they can dump all the red blood cells, and the red blood cells can be filtered. In some patients, there is something called like an accessory spleen, and it's usually still attached to the spleen, the original spleen itself. It's usually a like kind of benign condition. Okay, that's it for now. Thank you for your attention, and good luck studying.